want us to start? Okay. Do you want to open? You do. Welcome to the plenary session topic 6, special workshop on ethics, economics, big data, and artificial intelligence. I'm happy to co-chair this session with Dr. Joanna Namoado. Until recently, she headed in the EU Commission the ethics file, and now a consultant. Thank you.
And I'm very pleased to introduce Michal, who is a specialist in data and IT.
Thanks. And our first speaker, who's already standing, thank you very much, Matthias Pasternas, who is the data scientist, analytical development, cell and gene therapy catapult at United Kingdom. Thank you.
Thank you very much, and welcome back from your lunch break. Unsurprisingly, I will talk about data in the cell and gene therapy industry. And as a bit of an introduction, and as many of you will already be aware, cell and gene therapies have been quite successful in the last years. So we had some major breakthroughs with therapies reaching the market. And accordingly, there's a lot of excitement in that field. And we've seen a steep increase in the number of clinical trials. So we've seen a 32% increase from 2014 to 2018. However, it's still early days. It's still quite challenging to develop these therapies and to manufacture these therapies. And this might be one reason why we see eye-watering prices for those therapies that are actually already available on the market. So we see headline figures of up to $2.1 million per treatment and patient. And obviously this means that even though those therapies are available in theory, they are out of reach for many patients because they're simply priced out. And I would like to talk about how we can use data to tackle this issue and how we can use data to improve our understanding of those biological processes underpinning gene therapies, how we can use data to improve our manufacturing processes and increase the productivity and hopefully in the long term bring down those prices. If we think in terms of manufacturing environment, and as I said, it's quite expensive to do. And obviously, if you produce a therapy, in the end of that process, your batch will have to pass some kind of quality criteria. If you learn at the end of that expensive process that something has failed, that essentially means you have burned a lot of money until that point in time. Also, if you don't really understand why something has failed, you don't put yourself in a better position in the future in improving things. So what you really want to do is to understand what factors are involved and what factors are driving success and what factors driving failure. And thankfully, there's a whole range of different data types which we, in theory, could use to characterize biological processes. Traditionally, in the gene therapy field, people used methods such as flow cytometry and real-time qPCR, but the field is increasingly moving towards other data types such as transcriptomics, proteomics, metabolomics, spectroscopies, next generation sequencing, and integrated sensors. And these data types should really enable us to understand those biological processes in great detail, enabling us to understand what makes a good product and what makes a bad product. And once we understand those factors, we might be in a position to actually predict early on whether a product will be a success or a failure. So if we can, in that manufacturing process, if we can determine at an early point in time that something is bound to fail, we might be in a position to stop that manufacturing process early, helping us to save some money down the road. Better still, if we can have an adaptive control. So what we do at the Catapult is we're using Raman spectroscopy technology as a real-time monitoring tool to analyze the biological processes within a bioreactor. And what we envision is that we can use that signal from Raman spectroscopy to ascertain that something in the bioreactor is about to go wrong. And once we understood that something is about to go wrong, we can then use this adaptive control and change a parameter, say, for instance, the feeding regime. And then instead of discarding that batch at an early point in time, we might be able to rescue it and still saving more money, because this essentially means then we have to discard less batches in manufacturing. So that's all well and good, and as always in life, there are many challenges. One is that data sets tend to be fragmented, so we don't always have all that data. But in my personal view, as a data scientist and statistician, I think the biggest challenge is that in this field, we tend to work with really small data sets. So you can characterize your product, say, with proteomics or metabolomics, and this will give you loads of different measures, but you can't really do much with all those measures as long as you don't have the sample size to match the number of potential predictors. And at Catapult, we are tackling this issue by trying to automate as many processes as we can. This is concerning lab procedures itself, say by using liquid handling robots, but also by automating routine analysis tasks. And we believe that this will enable us to collect more high-quality data. And obviously, you can't just dump the data somewhere. You have to make sure that this data is findable and reusable and interoperable. And you should have a sound data strategy to start with. So you have to know what do I actually want, what are my questions, and then collect that data accordingly. But even if you have all those things in place, we believe that it would still be quite difficult for a single company to collect a sufficient amount of data. And obviously, this is a big data session. And I think in this field, we are struggling, or we're not really there yet. And at Catapult, we're trying to collaborate with other companies, and we're trying to foster that collaboration. And if we can bring different companies together and convince them to share some of that data and being in a position of having these large, big data sets, I think we could then really tackle some of those bottlenecks that we currently see, and which was then enable us to use some of those more advanced analytics and characterizing those biological processes in more detail, having more control over the development and manufacturing process, hopefully bringing down the development time, reducing the production costs, and all that should hopefully, in the long term, lead to decreased prices so that more patients have access to those life-saving treatments. With that, I would like to thank you for your attention. Do you have any questions?
Maybe I'll start with a question from here, Matthias, on the stage, because I don't see anyone yet asking. So Matthias, I would like to hear a success story. You gave the overall framework for decision support based on this very diverse data. Is there a concrete example that you can share with us where you used the methods of predictions in order to support generating decisions?
Yes, so we do have all those technologies in place, and we do generate these predictions. Still, we feel to really validate those statistical models in a more robust way would really benefit from having more data. So I think the challenge is not so much measuring that data, or at least we do that already, but having the scale in terms of data.
Thanks. Is there a question here?
No, it's an announcement, and it doesn't belong to your lecture.
Okay.
So many people ask me about the clips. They like it probably. So they are available in YouTube. And go ahead and use it. I just want to tell you about one of the artists that made it. He became so interested in structural biology that he left animation and became a professor in structural biology. Thank you.
Thanks a lot to Adayonat, Professor Adayonat, for the clarification. Any further questions? No further questions. I got the sign of no further question. Thank you very much.
The next speaker is Professor Andreas Kurz from the Charité University of Medicine and working in stem cell therapies and degenerative disease and ethics.
Okay, thank you very much, Joanna, for the introduction. I will talk about how to manage ethics, specifically in regenerative medicine and the topics we are dealing with. What do we mean with that? How is ethics as a discipline perceived? One perceives it as a lamenting group of people who see risks in every step we are taking, and the others saying, well, we need to be pragmatic about what we are doing. We are researchers, we can move ahead, and we want to move ahead as quick as we can. It is also putting roadblocks in front of people who say, well, this is what I want. I want this diesel truck. I want to drive it. I know it's maybe for the climate not the best thing to have, but I want to be safe in this truck. Or I want to do gene editing of my child because it will be smarter. We have the technology to do it. Why shouldn't I do it? Because I want it. So this imperative is quite common and perceived in the public as being a common trait among scientists. The truth is probably something in the negotiation between these two aspects, in putting roadblocks and moving forward science. So, in my opinion, ethics is so central for research. It makes research, puts research on a solid footing. And in sync, if it works well with the public, it's not a law. Ethics might result, ethic opinion might result in laws, but it's itself not a law, it's a guidance for behavior. It's not just for health, as I just mentioned, and it's not just paperwork. It is involving paperwork because you need to think, put your thoughts down and justify why you do a certain experiment or a certain clinical application or why you shouldn't do it. In such, it is a risk management tool, so you protect yourself with good ethics from encountering risks later on. It provides a regulatory conformity. You can get a paper trail of what you are doing. You can go back why this decision was made, what was the ethical consideration at that time, and it will generate confidence in not only you as a scientist, but science in general. So what does it mean to put ethics in the center of research in our area? I have listed a few aspects. One is animal research. So animal welfare, do we need animals if we have, for example, organs on our chip systems, which are mimicking better, perhaps, what we do in animals? How do we justify animal research? This experiment has been done before. Why should I do that again? With the same model. Human use and human welfare, of course, same as animal welfare, just for humans. There's gene editing, and how should we consider that gene editing and in the somatic aspect or in the germline aspect? Data usage, we just heard about that, has a very positive aspect when we want to optimize a treatment or a cell type. What do we do when we want to reuse data which has been collected 10 years ago without proper consent? How do we destruct data? Or shouldn't we do that if somebody wants their data to be removed from a public or research field? How do we handle cells and organoids which are banked, where the consent for that banking is, let's say, limited to a certain project? Last but not least, how do we handle the right to know of a donor of a tissue, for example, or not to know results of their genetic testing, for example? I will briefly go over two aspects. First one is gene editing, which has been stirred a lot of attention recently, and that's what you find in the news. Chinese created a monkey with superior mental power. You all heard about Professor He from China, who created the first genome-edited babies. And you can change basically species by simultaneously gene-edit 13,000 genes, and you don't care about any other side effect, because this is not the major point you want to make. You want to make a new species which is superior or fits much more your interest, your application. Yeah. So, and the question is then, so what is the road we want to take, right? How do we answer that? Why can't we eliminate disease in an embryo if we can? Why shouldn't we do that? Why shouldn't we alter genetic heritage, which is even reversible when we use genome editing? Why should people not enhance themselves and their children? Should the technology be available for all? This is an aspect of solidarity and fairness. Should it be or should it not be? And why not make human-like animals for drug screening or testing or understanding disease better? And how far can we go? So what do we know when we do genome editing? We think we know that how genomic information is transmitted, but do we really know? We are very close to that, I think, but there are many things we don't know. We don't know about the majority of genes, how they really work, and the complete interaction pattern in a cell or in an organ. What are the conditions, or what are the unintended consequences thus of this gene editing procedure? What changes in the genome might have unforeseen outcomes, and what is the effect on future generations? Is the attempt to reshape species in our current state of knowledge acceptable, or is it not acceptable? Where are the limits? And how do we decide these limits? What is safe enough? And I don't know whether you can read that, but this wolf says, well, this human has a fire, has food, why not go there? What can happen possibly? And 20,000 years later, we know what happens to that animal. So there's a lot of studies about perceived risk, how to assess risks with a limited knowledge about that risk. And the answer will probably be, it depends. It depends what is safe enough on the application we want to do. And this is where ethics comes into play. There's always a risk-benefit evaluation, and we will see what then can be done, what should be done, and what should perhaps not be done. There is a moratorium put out by some scientists on genome editing and germline genome editing, which is a voluntary moratorium, but it's probably unlikely to stop rock scientists, right, who still want to do it. I want to do it, I can do it. And some people feel that's too rigid, especially because there are no defined ways out of this moratorium. So what should be the condition to say, well, this moratorium is now over and we can do genome editing in the baby? We know the Hill experiment. China is now putting a very strict, or very, let's say, we don't know it yet, a new regulation into place, and there are guidelines to control the spread of genome editing in babies under and allow it under very specific conditions. So next topic, very briefly, I want to talk about data. So when we are going to the doctor, we trust the doctor that the data he has to evaluate us are correct. But as a citizen, very often people say, well, I don't want to give you my data for public use because I'm fearing that these data are misused, that the data are not private anymore, and so on. So how can we balance these two aspects? So use of data and for the public good and protection of the privacy. Are there only two ways to go, or is there something in between to make both possible? And at the moment, I think the solutions are not clear to many people. Although in Europe, we all know that data protection regulation has been put into place, which enables both data usage and protection of privacy. And I will very quickly go over that because the time is already over, and go to the last part, which is management and governance. So we should balance the data, consent, and privacy. As you know, we must identify best practice to handle data, even if they're not anonymizable, for example, genetic data, how to handle this and make them available for the public. So these are the regulations which are already in place, in Europe at least, to handle genome editing. And there was a talk before to discuss, well, is the regulation sufficient, which we already have, or what should be modified? And I mentioned the data protection regulation already. So in conclusion, ethics isn't morals, it's not the law, it's a lever of compliance. And it's not a defense against liability. You are liable anyway, even if you are ethical. And it's a keeper of documents. We need to document your ethical considerations, and you can trace them. And then for next time you have the same ethical question, you can go back to that. And it's a guidance for the researcher on the bench or the doctor on the bed. Right? Then they often don't know, is that what I'm doing ethical, or or should I not do that perhaps? This is the level beyond or below the meta level of ethical considerations of naturalness, solidarity, freedom, dignity, and so on. How useful is that for the researcher and for the medical practitioner? So, and with that, I stop. I leave that to read, and thank you very much for your attention.
We'll leave the questions for the next time. I would like to introduce the next speaker, Pier Maria Fornazzari, who is interested in ethics and research communication.
Grazie. Thank you to Restore Community for the invitation to this very important meeting. And in order to avoid overlaps, I was asked to present the topic of ethics of stem cell therapies information in a digital area through social media. And I will try to explain it with three outlooks. The first one is the social media role is in stem cell therapies in the digital era. And the second one is unapproved stem cell therapies clinics diffusion, the role of social media. And the third one is what we can do in correct information dissemination. In this year, the global number of internet users has surpassed 4 billion. And there is an average internet user spending around 6.5 hours per day using devices connected to internet. And similarly, 3.5 billion people use some type of social media platform with an increase of 1 billion over the past year. This is very important because it's clearly a market largely greater than the newspapers and other forms of communications. And for looking at the research of Pew Center, 80% of internet users have searched for health-related topic online, and most frequently, people went online to look up information, 63% about a specific disease, and 47% about a particular medical treatment. So what and why internet users are searching about stem cells therapies? This is a paper published last year by a great group of scientists under the edge of Lancet. And they say that the popular situation is that there is popular optimism and excitement about the potential impact of regenerative medicine. But there is a gap between people's expectation that new therapy should be available and the realities of translating them into the clinical practice. And where novel therapies involve patients who have no other option for treatment and are desperate, the hope of a cure can make them highly vulnerable to wishful thinking, and where money is involved, vulnerable to false promises. So the problem is the hope. Is the role of the hope which creates or sustaining false hope, and this is ethically problematic. And this has brought to the growth, international growth of purely regulated or unregulated clinic that appeal to disparate patients and their families using social media. And this is the second point. Purely regulated clinics published in the papers, you find that are more than 1,000 all over the world, but probably are even more. And they're present in USA, in Canada, in Australia, in Japan, Germany, and UK, following these papers by Turner and Knöpfler. And this type of market, which is called direct-to-marketing, creates a continuous travels of patients from and to different countries. For example, here it's published that the patient come for 65% from US, 11 from India, 7 from Canada, and travels to Panama, Mexico, India, US, etc. The second point is how these providers diffuse their idea, and often they use patient testimonials. The role of patient testimonials is very important because internet users have been shown to identify more strongly with audio-video testimonials as opposed to text or picture-based testimonials. And in these videos, patients mentioned risks in 10% and benefits in 95%. For example, this video made by Mel Gibson discussing the therapies done to his father in Panama had a market of over 2 million views. Other unwilling testimonials are all the athletes which use or stem cells or PRP or other regenerative therapies, and it's well known that they use. And so, says James Rickert, which is the president of the Society for Patient-Centered Orthopedics, this sends a signal to all the fans out there that stem cells have more value than they really do. So, as the benefits are published, are highlighted, there are very few data about adverse event. This is the only one paper published by a group of a friend, Mohamed Abou El-Enain, and they found only 35 cases, 19 from scientific literatures, and 16 were mass media reports. So the number is effectively very, very low, and this is another problem. What we can do, always this group says that the solution lies in a coordinated strategies with 4 pillars: better science, better funding models, better governance, and better public and patient engagement. I highlight only this last one, of course. And so we want to communicate to circumvent misinformation, to educate and navigate patients, and we have to share valuable content. People need a reason to follow and engage with us, and the information need to be credible, of course, and true, but also creative and entertaining. So if you want to use, and we have to use social media to explain people, to educate the public, we have to ask and to highlight what is the core problem for me. Is epistemic authority science effective in communication to patients? And this is the point which is published by a series of authors. The digital era is characterized by growing skepticism toward conventional scientific medicine. Top-down forms of medical knowledge are becoming increasingly contested. Actors outside the academy and the medical profession, including patients and consumers, occupy increasingly active roles in the production and interpretation of scientific and health knowledge. And the last one, which is the most important, it can no longer be presumed that the public respect for facts can be secured via the authority of expert legitimated by agency outside the public domain. This is an article of Marres. In fact, we have also to highlight that the DTC website deploy a future-oriented rhetoric where scientific experimentation is justified by the promise of a new and better treatments. Consumers can position themselves as front runners of science. And so the promissory depiction is in relation to a new era of regenerative medicine, and consumers are given the opportunity to be a part of realizing this new era. So we have to discuss what is our position, our epistemic authority position in this new era. What we can do again? For example, we can make some regenerative medicine consultation services, like has been made by Mayo Clinic, which is a clinical service where people can phone, can discuss with the doctors of the group, and have conversation consultant, patient medical evaluation, and correcting misinformation. Another possibility is to diffuse fake news checking instruments. This is one, for example, published on Scientific American. This is another one. Another point is increased regulatory enforcement is necessary. Yes, but is not sufficient. Civil litigation efforts are not sufficient, but they need also an increased public education campaigns. For example, this is an article published by Paul Knöpfler this morning, yesterday. FDA letter was the agency's long game. What is doing FDA to stop and how is effective in it? The last one is the possibility for Horizon grants. For example, this one is very interesting. Is closing at 7 April next year, and is reliable and accessible information on cell and gene-based therapies. And I think that we can take part to it. Here I have put some working examples very quickly. One is the Mayo Clinic minutes. The other one is FDA, which released a 1-minute video. Then there is our website Restore, Regenerate Solution Restore, the niche, very authoritative by Paul Knöpfler. And this very interesting podcast is Bad Batch. Is Laura Bales made like a story in 6-7 parts, looking at the fraud of a society which sells stem cells. Thank you.
Thank you very much, Pier Maria. Again, with an ethics talk, we have no time for questions at this precise moment. Perhaps we will gather some time later on and pitch you some questions. I have the pleasure of introducing now Michael Rosenzweig, who is Director of Health Informatics, with a particular interest in machine learning and artificial intelligence.
Hi. I will start with defining machine learning in the context of AI, because I want to make sure that we all talk about the exact same thing. And I hope to take you in the next 10 minutes through the potential value of AI in the context of drug development and treatment development, and then to provide some example of where we used it, and to finish with some open requests, open opportunities, and open challenges. And I'll start by referring to Professor Michael Jordan, who is not the basketball player you might have in mind. Professor Michael Jordan from UC Berkeley, who was my advisor when I did my postdoc, talks about what machine learning is. And he is illustrating machine learning as some kind of a wedding between computer science and statistics. Because when you want to analyze data and to use advanced statistics, you can create very complex models. But if you cannot analyze these models because there's no algorithm that in feasible time can infer aspects from these models, then is useless. So when you create analysis of data through statistics while keeping in mind how an algorithm can resolve that, that would be machine learning, or one way to define machine learning. And Professor Jordan and Professor Tom Mitchell wrote a paper in Science about the trends in machine learning and where machine learning goes. And they talk about what are the open challenges and the things that still the community is working on. And in order to be very concrete and to simplify the world the way physicists are doing, and my training is as a physicist, I'll take the very simple example that many of the people in the room here are familiar with, which is the case where you have a condition and a novel, very exciting advanced therapy that you can offer for that condition. Now, in this very simple example, in the condition, there are a vast majority of the people who can be well treated, and small portion of the people who have complications, and this small subgroup has much more complicated disease, and they need advanced therapy. Now, if you look retrospectively at data where most of them were treated with classic treatment and were cured, some of them were treated with advanced therapy and were not cured, and all the most challenging people who were treated with the advanced therapy, or almost all of them, might have been benefited and cured from that or had improvements. When you look at such data retrospectively in this very simple example, people will say advanced therapy is not good because most of the cases for this condition were cured without the advanced therapy. And that's, of course, because if you only look at the data without taking into account the severity of the condition, then on the face of it, it looks like that. It looks, using this very simplistic modeling, it looks as if the prediction will be that advanced therapy is a sign for not being able to cure, if you want to do a prediction. And also, as a prediction, it might be correct. If you don't know anything about a person, you just can look if they obtained advanced therapy. Probably they are in the very severe aspect of the spectrum of the condition, and they might not be cured. But that's the big difference between prediction and causal inference. Causal inference means that you don't only look at the prediction model, you also try to understand the cause and effect. And if in the prediction story, very simple one that I shared before, if in the prediction model we have understood that there's this another layer of information that is of importance, which is the severity, then the prediction would have been more accurate, and then the conclusion wouldn't be a mistake. Then if we had the information about the severity and we could take it into account in our model, then prediction model would be more accurate, and we will not jump to the wrong conclusion that advanced therapy doesn't help. But in many cases, the story is much more complex, and the many features appearing are much more diverse, and not always it is recorded. So understanding scalable causality is something, and that's the first item here, prediction versus causality, is something that the community is developing. And I would like to claim that for Restore, for the needs of this consortium where you develop advanced therapies and you want to understand cause and effect retrospectively from data, and you can benefit from that, usage of this technology is important, and development of such a technology is important. Of course, and here I'm doing another assumption of what you have in your mind. If I ask you, what's the most ancient profession in the world? I don't know what you think, but I would say it's medicine, because we all know that giving a hug to someone who just fell, to our kid who just fell, is some kind of a medicine, and I can imagine the first parents taking care of the kids in this way. So medicine is something that is very ancient, and knowledge was accumulated over the years. And machine learning, the way it was defined before, statistical models that learn from data, that takes into account computer science and how to generate an algorithm out of it, doesn't take into account in a natural way the knowledge that you have. But advanced successful machine learning in the context of healthcare do need to take into account all the medical knowledge. And there are different methods and ways to incorporate knowledge in algorithms. And that's also a key component, in my view, in order to progress medicine and to provide insights with respect to advanced therapy using machine learning. Never-ending learner is another aspect of learning that is not yet resolved at all, but I don't want to spend more time on that because we have very limited time. And I want to emphasize that in order to use the benefits, to enjoy the value of AI, as was presented before by Matthias in the beginning of this session, we need access to data. And of course, in the care systems, in the providers, the payers, and the operating systems across the world, there's a lot of data. And this data is being collected, and this data is being leveraged, as I will share in the specific example. And also within Europe, there are different efforts to benefit from that data, from the data that is collected and managed within the different hospitals. An example is the Big Medalytics project, which is the largest EU-funded initiative to transform the region's healthcare sector by using state-of-the-art big data technologies. And we are part of that, and we believe that's a venue and a way to benefit from the data that exists in Europe and to explore with that. Of course, there are many different aspects, ethics and so on, that are discussed today. These are also addressed through this project, the Big Medalytics. Now I'm going to focus on drug development related issues, and I want to start with highlighting a paper that just went out in Trends in Pharmacological Sciences. And in this paper, and I don't think I have sufficient time to take into account and explore all the aspects, I want to focus on a few of them, but this paper maps out all the opportunities of AI in the context of drug development. And when you think about the design of a study, you can think of how to create eligibility criteria and to develop the eligibility criteria in such a way that it will provide the most likelihood of success to the system. And that's one way to use and benefit from AI. There are many other ways, and I'm not going to review all of that. Stefan Harrer and other people here shared their view on this, and it's a nice, very good review paper. The example I want to share that in the context of drug development is the opportunity to benefit from retrospective data when you want to find new indications for existing drugs. When you want to find new indications for existing drugs, you want to leapfrog and to accelerate the development of drugs such that you don't need to establish safety, the phase 1, because you already have that these are drugs that are in the market, and you might need to do some exploration for the efficacy. But first indication for efficacy might be already there in the data. And I would like to share results that we are about to publish very soon of a study we performed with Teva, a generic pharmaceutical company, that when we leveraged AI technologies and access to data that we have through data sets in the US that all in all had more than or an order of 180 million individuals in the data, and we looked into Parkinson, we were looking retrospectively, can we create a situation that looks like a clinical trial, that takes into account causality and goes beyond prediction, that evaluates who obtained the drugs and why, so that we can compare apples to apples when we take a group who used the drug and a certain drug and a group that did not use the same drug, and to show that that drug might have benefits not known before. I'm not going to elaborate more about the two results that were found out. These two results are found, the Resadgelin and the Zolpidem, were two drugs that were found to be of value for the dementia as an outcome of Parkinson. And this is only based on the data analysis with some knowledge incorporation. More details, I'm happy to answer questions in the break. And I'll finish with, while prediction modeling is easy to test, you put aside held out data and you test on it. Causal inference is much harder to test and evaluate. And that's why we created a tool that is open sourced and people can use it for benchmarking their ability to generate stable causal inference. Thank you very much. Do we have time for questions? Yes. Are there any questions?
Thank you very much for this nice talk. My question is, one problem in advanced therapies is that you have normally very low numbers of patients in clinical trials, both in the phase 1, mostly have 3+3 design or whatever. But even in the approval trials, because of the high efficiency, you have maximum 100 patients or whatever, which is of course not comparable with the conventional pharma development where you have hundreds or even thousands of patients. Do you think that the number of, or the data from such low number of patients is sufficient to generate such kinds of tools you mentioned here?
Yeah, that's a very important question that we try to deal with. And of course, there are challenges because when you use statistics, you need to have sufficiently large cohort to generate statistically significant results. So it's a very good question. And then the answer lies in the details of if you have a certain condition and you have people who suffer from that condition from whom you have large quantities of data because they are treated in the regular way and they are in the system. And then you compare and generate insights from this large cohort and just test and apply them to the small cohort, then some conclusions can be drawn. And there is a recent study, I didn't bring it here, I actually should have. There's a recent study that shows how many of the clinical trials can be repeated through retrospective studies. I think it was 70%, which sounds low on one hand, but on the other hand, if you design upfront the advanced therapy study in such a way that you want to make sure that you will have more information in your cohort, so you design the endpoints in a way that they match what you know you have in your cohort patients' data, and then you increase the likelihood to benefit from the cohort patients' data. And this requires advanced planning. So I think it's a good question and it's an open question. I think there are mitigations to the small data, but it's an open exploration. We actually are now looking at this very question.
I also have a question in this context, actually. So maybe you already answered part of that, but we design all our studies now not with the prospect of being retrospectively analyzed, our prospective studies, by a large AI effort then. Do we have to bear this in mind in all future studies? And where this 70% that you now said, was this the 70% of all studies we can analyze retrospectively, like for the Parkinson's and dementia patients?
So the question, I'm not sure.
So the question is concerning all the data protection laws actually that we are running in. We have to dedicate our data and we have to say what we want to do with our data. And the AI is not yet a part of the whole thing. So all the trials that are running are actually not meant to be analyzed retrospectively by an AI algorithm. So is this a problem, or can we already take this data of all the trials that we have already done and let the AI go over them?
That's very complex, and as you said, I will not give a full answer to that. I can share some of our experience. In the past, we worked on HIV data, and we had access to 65,000 individuals' data on HIV, and also some access to trial data. Since the commonalities, and the trial data was prospective, not designed to be analyzed in the context of retrospective data. It was a specific clinical trial. But as we were focused on a very specific aspect of optimizing the treatment based on the virus mutations, and the virus mutations that were collected, the information was collected in the clinical trial. And we took into account the bias that was in the eligibility criteria of the clinical trial, we were able to benefit and to increase our accuracy based on that clinical trial. We ruled out other clinical trials, so it depends a lot on the details of the prospective studies. I believe that we can benefit from linking them to cohort data, but it requires sitting on each and every aspect and making this effort. Thanks.
Thank you very much, Michal. I would like to introduce Lisbeth Garris, Professor of Biomechanics and Computational Tissue Engineering, with a particular interest in silico medicine and systems biology.
Thank you very much. Thank you, Michael, for the very great introduction. I would like to continue and expand the toolbox a little bit from the AI to wider examples and also to other applications other than the clinical aspects of it. So first and foremost, I would like to thank my team who developed all these examples that I will be showing. So the context of this particular talk is tissue engineering. We've talked now a lot about cell and gene therapies. This is cell-based tissue engineering strategies. And in the platform that we developed, we work in the context of bone, and we work along all the phases of the developmental process, which means that we start with looking into cells and growth factors and materials. We combine them to look into mechanisms of action when we implant them. Then once we know what we're doing, or we think we know what we're doing, we're trying to upscale that. So we're also working on the process engineering aspects, looking at bioreactors, looking at the large animal experiments. And then ultimately, we're now at the stage that we're interacting with the regulators to prepare our first clinical trial next year. Now, I'm not going to talk about these particular applications. What I want to focus on now is on the underlying thing that we're developing, and those are the enabling technologies. So the technologies that allow us to answer questions all along this pipeline. Not one model, and the specific thing that I want to talk about is the modeling aspect. And the models that we're talking about is not just one model for the whole thing, but specific models to answer specific questions in a specific context of use. And these models are always done in an integrative loop, so meaning hand in hand with the experimental work and the questions and the biological questions underneath it. Now, in silico is a spectrum. We've heard a lot about the data-driven side, so the black box side, if you want, which, of course, black box is one aspect. AI is more than just a black box, but those could be considered synonyms by some. On the other side, if you want to be black and white, you have the white box models where you start from first principles, the mechanistic models. If you know how something works, or you think you know how something works, you can translate that into mathematical models, and you could use that to try and get an answer to your question. But more often than not, the truth is somewhere in the middle, just the same way as we try to incorporate mechanisms in the data-driven approaches. We also need data-driven approaches to fill out the blanks that we have in our mechanistic approaches. So in the end, we will end up, also due to the fact that we have little data or not large enough data sets, we will probably need to use some combination of both approaches. So what I want to do now in the minutes that rest me, and probably I will run out of time in the end, so I'm happy to answer any questions during a coffee break, is look at some examples how we use these things in our platform in the lab. Starting with, for instance, things that we can do to look at simple questions that we have with the cells. One of the building blocks, for instance, that we use in the lab are not individual cells, but little aggregates of cells. And the question is then, well, you can make these aggregates as big as or as small as you want them. So what would be the optimal size of these aggregates? Well, rather than doing a full study, you can look at computational aspects. For instance, if we want to have little aggregates where we don't have any gradients, well, what is then the size of the aggregate that you need to use? So if we look here at a model of this aggregate, you can see that you can simulate for these different aggregates. There's the pointer. You can simulate different sizes, and you can decide based on the model where you don't have a gradient and then use that size. A very simple example. Another example is to use all the data-driven approaches that were already mentioned by the first speaker. So you can look at bulk RNA sequencing, single-cell RNA sequencing, metabolomics to get a better understanding of everything that you're trying to build in the lab, and then use that in some way as a quality control when you're going to produce this at a larger scale later on. Now, from all these insights, you can also generate data, you can also generate networks. And these networks you can use in a systems biology approach. You can look at dynamic behavior of such networks. If you think, well, this is the core network of the biology that I'm studying, well, then what can you do? You can take such a network and you can try to look at, well, based on this network, what would be the most likely outcome of my cell states that I'm using or that I'm differentiating. And if I know what are my cell states, then how can I manipulate that? Because that's what we want to do in the lab. We want to use a combination of factors in the medium to try and push the cells into a certain direction. So how can we use all of that knowledge to try and predict or do an in silico screening before we actually do the in vitro screening? Now, If we go to carriers, well, there again, you have a lot of experiments that have been done, and from that experimental data, you can do some empirical derivation of what would be most important aspects of, in this case, calcium phosphate-based scaffolds. But there is also another approach where you can start from a mechanism. When you look at scaffolds and how cells behave inside scaffolds when you culture them, you see that cells like to go sit in corners. So they're always attracted to these curvatures. Curvature-based biology is something that is really up and coming. And if you use that as a basis for a mechanistic model, well, you can actually simulate something that is both qualitatively and quantitatively very similar to what you get from the experiment. So this can be used as a baseline for developing scaffold shapes that would presumably lead to a better behavior. So one of these things that we did is where we compared a gyroid-based structure that we developed based on the models, and we compared that with the golden standard material, the BIOS, and a lattice-based structure that is also relatively frequently used. And what we saw after 4 weeks, not any particular differences, but after 8 weeks, you really started to see differences in the amount of bone that was formed in this rat model. And what was actually equally important as the amount that is formed is the fact that here you see the darker gray, and that is the bone that was reaching all the way up to the top of the implant, whereas for the other scaffolds, it always stayed at the baseline close to the skull. So using insights in biology, you can leverage that to design a better structure for your biomaterial. Now, once you have these cells and once you have these biomaterials, you need to culture them, or sometimes you want to include a culturing step. So if you look, for instance, at perfusion bioreactor studies, well, then what you want to do is try to understand what happens to the bioreactor that you have, where you have the inflow part where you have sensors, and then you have the outflow where you have another bunch of sensors. And we want to model what is happening inside this bioreactor. Now, I'm not going to talk about what is in the model. It's basically, again, this curvature-based growth combined with the effects of oxygen and glucose and lactate on the cells and the shear stress induced by the fluid flow. And here, just showing 2 examples of what then happens, where you place the scaffold at the bottom of the bioreactor chamber. You see that you get very poor filling here at the bottom, at the inlet, because the flow is not well developed. Whereas if you place your scaffold in the middle, which anybody who works with bioreactors already would know, is that you get a much more developed flow and you get a much more homogeneous scaffold filling. Now, with this kind of model, you can then start playing. You can look at optimized scaffolds for your bioreactor or your cell culture. You can look at optimized culture strategies for reaching, for instance, maximum filling of your scaffold or other outcomes that you can define. And ultimately, what we're now doing is including this model inside the monitoring control loop so that we actually have a digital twin for a bioreactor that we can optimize the process and that we can automate the process. Now, looking at clinical applications, again, we're working, the patient population that we're working for is patients or children with neurofibromatosis, which is an orthopedic and an orphan, which is a pediatric and an orphan indication. So there is very little data available, only case studies. But what we do know from biology is what are the processes that are going wrong in the bone formation. And so we can include that in a mechanistic model of normal fracture healing, and we can adapt that. We can then build, instead of having real patients, we can build 200 patients that have neurofibromatosis type of bone formation. And then we can run in silico clinical studies where we have here patients that have not been treated with good healing for low values, abnormal healing for high values, and do the same when they have been given a treatment. So you get 4 patient categories if you then take that data and use machine learning or classifiers to look at what types of patients that you have. Patients that are healing, patients that are responding, patients that are not responders, and then patients that are adverse responders. So you can use this in silico data and then do machine learning on that. But why would you believe, and I'm almost done, why would you believe now anything that we're saying? So how can we establish credibility? If I go to a regulator, why would they accept the evidence that I have here? Well, we first have to document, and FDA has already provided a lot of documents there how to do that. And then we have to establish the VVUQ. So we have to show that our models correspond to the mathematics. We have to show that our simulation results correspond to the physical reality. And then we have to show how the assumptions that we made, the impact of that affects the validity of the simulation results in the end. So do uncertainty quantification. And this then has now led into a standard for doing verification and validation, which has been established by ASME and FDA. So this now really is something that they accept. And in the European context, we're trying to get that done. We now are at the stage that in the MDR and the EMA legislation, it is explicitly mentioned that they have to accept digital evidence if we provide it to them. But now they have to establish ways of validating, of assessing this evidence. And I'd like to end with this slide, which is a slide from the FDA showing that currently, or actually a few years ago, most of the data in their regulatory filing was experimental and in vivo. While in the future, they predict that almost half of that information will come from in silico clinical trials. And just last year, they approved the first clinical trial where all of the data or where all of the clinical studies was replaced by an in silico clinical trial. Thank you very much for your attention.
Thanks. As you correctly predicted, no time for questions. Thank you very much for the interesting presentation. I'm happy to invite Professor Andreas Kurz from Charité University here in Berlin, Germany, to talk about stem cell data standards and data utility.
Yeah, it's me again. Thank you. Sorry about that. Yeah, I want to talk about the, data on data standards, specifically on cells. And I focus on induced pluripotent stem cells. There was a question on well, small cohorts in advanced therapy applications, patient data based on these small cohorts. But the other side of the spectrum, especially for cell therapy, is the cell itself and what kind of data we have here and can we collect. So there is a, you know, that a huge data challenge, and we get data every day on the, so soon there will be 10 to the 10 people living on the world with 10 to the 5 diseases, and we have about 30,000 coding genes, protein coding genes, 10 to the 3 cell types, depending on whom you ask, and at least 100 or maybe about 100 organs. And in the moment, we have about 10,000 human iPS cell lines worldwide available, in theory being genetic surrogates of these 10 to the 10 people. So, and when I choose the iPS cells to demonstrate the need for data standards, because this covers a whole spectrum of data. It's going from the patient through the reprogramming and culturing procedure to the cell itself. So what is the characteristics of these pluripotent stem cells? The cells derived from the pluripotent stem cells, including organoids or single cells, single specific cell types, and then data on the application, either in disease models or in a clinical application. And again with iPS cells, there are very few patients usually enrolled in these clinical trials. So I would like to coin the term digital stem cell technologies. And this is basically summarizing what you saw before. We have the disease cohort data, you have the data on the iPS cells, the translational data in a way, on gene editing of these cells, on the differentiated cells, and then on the disease models. And you would like to have all these data together to make predictions, for example, to use AI on a data set which you can trust. This trustable data set depends a lot on the quality and on the comparability, interoperability of the data. You want to compare the data. So first of all, they need to be findable. So the data should be, if possible, globally visible, especially when it comes to cells. They should be findable, accessible, and they need to be somehow transparent and interoperable. And to make them interoperable, the easiest way to do that is to use standards. And I will just use one example. There's a standard nomenclature for the date, which is in the middle, 2013-02-27 is a standard, but this is not often used, right? There are many other ways to write a date, and if you now look what happens on a specific date in the World Wide Web, you will not find it if you use a standard name for a date. You need to ideally to use all the synonyms for this state. So this is hampering interoperability of information. And the other aspect is representation. So what file formats are we using to represent a certain set of data? I will talk a little bit about a registry for pluripotent stem cells, which we developed. And here you encounter especially exactly this problem, that the data need to be interoperable, they need to be findable and accessible. So we developed a standard identifier for pluripotent stem cell lines. This is the identifier. It is linked to other identifiers, for example, biosamples ID, which are used by EBI or NIH or RIKEN, and RR-ID. So research IDs, which are now becoming more and more common for all kinds of research-related data. So even if you use a different name than that, linkage is important, and that you then identify, well, this is linked to this particular standard identifier, and I can use it. So what kind of data do we collect on pluripotent stem cell lines? And some are listed here. We need, of course, contact information, who took the samples from the donor, who owns the cells, who generated the cells, who distributes these cells. We need the ethics information. So what was the ethics behind, or the consent behind donating these cells? We, of course, need to provide a unique identifier. We need information on the disease status of the donor, right? And here the problems already start. We need, of course, to know what cell type is it, and which the donor donated, and what cell type has been derived from that pluripotent stem cell lines, and how useful is it? And then there are other data like potency, identity, cell line identity. So is it exactly from this donor? Is the SGR or HLA profile identical? And the derivation information. So what are the standardization questions? First is there actually a need for standards? Do we really need to standardize all data which are associated with a cell type? And that depends really on the utilization. What are possible use cases? I will go to one later on. What are the technical criteria and data formats which are useful and which can be really implemented in the science and in the science lab? What should the model for a cell look like actually? And this is an issue discussed by many people who are working on the Human Cell Atlas and similar projects. What are the ethical standards to follow? So here's a use case: find, share, and analyze data. There are clinical data, right, from a patient, and the data standards here are very diverse. And there are genetic data, for example, where the problem is access, because genetic data are private data. We need a certain access module to access them. And then you want to combine them to work with them. And here, I think standards need to be developed for clinical data. This is access to data is usually regulated by a data access committee. So this is quite common. Genetic data are archived somewhere. Access is regulated by a data access committee, and I'm finishing in a few minutes. How to standardize experimental data. This is the hard part and not really solved. In the moment, at least we in the registry for pluripotent stem cell accept all kinds of data to make it really transparent. However, this is very difficult to search. And one kind of standard we use is ontologies, and this is just an example. People enter free text to describe a cell type or a donor disease, and we then search for a corresponding ontology term, which is standardized, and this term is then used as an ID for that particular characteristics or data. This is an example for this ontology terms. We use an EBI instrument called Zuma, which searches all kinds of ontologies to find the correct term. I don't want to go into that because the time is running. I just want to mention one thing. This is When you go to the registry website, you see that you can register your cells and your data, and you can use standardized terms. This is one step, and then you want to make cells offset, and you want to know how close these cells are to the cells in the organism. And for that, there is a data model being developed, which allows you to zoom into a human body, in this case, a kidney here, and you can assign, for example, gene expression data to that particular organ or organ element. And for that, to do that, you need data standards. And for genetic data, standards are available, but they are very difficult. Many people don't understand this nomenclature. And I think with that, this is my last slide. So we have also in the registry other data, for example, the clinical trial database. And the goal is, of course, to use this data from the clinical trial database for pluripotent stem cells and the cell type data or the IPS data to eventually, when enough data are there and they are standardized and interoperable, make predictions. With that, I would like to close. Thank you very much.
Thank you very much. We don't have time for questions. Very interesting session. I hope we can all interrogate the speakers during the coffee break. Feel free to grill them.